name is Nazar Vassiliou. I am brain surgeon and CEO of Global Alliance for Neurosurgical Brain Cancer Research Innovations. I'm delighted to share the latest publication targeting glioblastoma inducing necroptosis molecular pathway post-photodynamic therapy for enhanced neurosurgical and therapeutic outcomes, which is now available on ResearchGate. The research investigates the role of photodynamic therapy in activating the necroptosis pathway in glioblastoma cells, aiming to improve the efficacy of surgical and therapeutic approaches for this aggressive brain cancer. By enhancing cell death through necroptosis, photodynamic therapy holds promise for advancing treatment strategies and improving patient outcomes. Just a brief overview. According to the World Health Organization, glioblastoma is classified as fourth stage of brain cancer with lethal prognosis and survival rate of less than 5 years following the standard of care. Only 3% of affected patients survive beyond 5 years. Despite surgery, chemotherapy, and fraction therapy, tumors invariably recur, and it is important to note that radiotherapy presents several challenges, including the need for precise targeting to minimize damage to surrounding healthy tissue and management of potential side effects, such as radiation-induced toxicity. Despite all these challenges and development of resistance in cancer cells, over time, radiotherapy remains an integral part of multimodal treatment approach for GBM. 5-LA in combination with PDT, which converts to protoporphyrin IX, is widely used in neurosurgical resections of GBM as well as in research. It can be activated with wavelengths of 415 up to 635 nanometers wavelength for PDT, and it makes 5-LA an ideal photosynthesizer for routine and clinical practice at the moment and research as well. In this research, 100 micromolar concentration of 5-LA was used, and it was combined with PDT. According to different scientific sources, concentration of 100 micromolar was equivalent to the concentration of protoporphyrin IX in tumor brain tissue. However, different concentration of 5-LA has been tested in various in vitro and in vivo studies in combination with PDT, which resulted in cell death in the past. So the main goal of this research was to understand the molecular pathways involved in post-PDT5-LA induced cell death in glioblastoma stem cells. To delve deeper into the cell death phenomenon, molecular pathways were analyzed and are shown by demonstrating the main differences between the apoptosis and necroptosis cell death pathways, where two major cell deaths occur independently. The molecular pathway of necroptosis involves specific signaling molecules and steps in which cellular response-induced stress can lead to activation of necroptotic signaling pathways. This activation can be mediated by the presence of specific cellular receptors, such as death receptors, such as tumor necrosis factor receptor 1 and toll-like receptors, or through other mechanisms involving mitochondrial dysfunction and intracellular signaling. In the presence of death receptor activation such as TNFR1 by binding of ligands on and such as tumor necrosis factor alpha triggers the formation of a complex called the necrosome. The necrosome comprises of receptor-interacting protein kinase 1, receptor-interacting protein kinase 3, and mixed lineage kinase-like domain. Upon ligand binding, RIPK1 undergoes post-translational modifications, including ubiquitination, which can determine the signaling outcomes. During necroptosis, RIPK1 is phosphorylated, which is important because this promotes the recruitment of activation of RIPK3. Activated RIPK3 in order undergoes after phosphorylation and phosphorylates other RIPK3 molecules. Phosphorylated MLKL forms oligomers and translocates to the plasma membrane, where it disrupts membrane integrity. This disruption leads to leakage of intracellular contents, ultimately resulting in cell lysis and necrotic cell death. Next, to address the inquiry regarding the cytotoxic effects of post-PDT5LA treatment in glioblastoma stem cells, Opera Phoenix robotic analysis was conducted. This assay employed cytox green fluorescence labeling to distinguish dying cells within the total cell count marked with nuclear staining DAPI. Upon binding to the DNA of deceased cells, cytox green elicited a discernible green fluorescence signal, thereby facilitating the identification and quantification of cell death events. So in this figure, photodynamic therapy at 390 joules centimeter square plus 5LA demonstrated a significant increase in cell death in time-dependent manner, and approximately 80% of cell death were detected at 80 minutes after exposure to PDT in G7 GSCs. The heat map represents the mean of 3 patient-derived glioblastoma stem cells, the time 20, 40, 60, and 80 minutes respectively after the fluence rate, which demonstrated the percentage of cell death after exposure to PDT. And two-way ANOVA analysis was conducted in these 3 cell lines, which revealed significant cell death across the specific time points, with a notable difference observed between treatment groups: DMSO light alone, 5 Vala alone versus 390 joules centimeter square for 15 nanometers plus photodynamic therapy plus 5LA. While GSCs are superpopulation of cancer cells within gliomas that possess stem cell-like properties such as cell renewal and differentiation capacity, the clonogenic assay assesses the ability of single cells to form colonies, providing a measure of long-term cell survival. Experiments were conducted to determine whether GSCs can lose their stemness after exposure to PDT5LA at different fluence rates, represented in joules centimeter square over a period of 10 days following illumination. In summary, 390 joules centimeter square and 160 joules centimeter square clinically relevant fluence rate demonstrated potent cytotoxic effect after PDT5LA, and no colonies were seen. Next, to delve deep into cell death mechanisms, cell death inhibitors were used, such as necroptosis inhibitor, which is seen here, necrosulfonamide, and caspase-3 inhibitors at depth FMK. These inhibitors can assist in elucidating specific cell death pathways involved in response to PDT5LA treatment. A little summary and effectiveness of the DEVT-FMK inhibiting G7 GSCs post-PDT coupled with immediate cell demise and suggested non-apoptotic mode of cell death predominance. These outcomes underscores the likelihood of alternative cell death pathways potentially implicated necroptosis and other mediated mechanisms, and thereby illustrating the intricate cellular response to PDT treatment in G7 GSCs. Next, Incasite experiment, which is depicted and illustrates the presence of G7 glioblastoma stem cells to PDT5LA treatment with and without NSA from 3 biologically independent experiments. By the way, the percentage of cytox green positive cells were calculated from total cell count, and values were normalized with G7 GCs treated with 10 micromole of staurosporin. Two-way ANOVA analysis if alpha was more than 0.05 and statistical significance seen between photodynamic therapy 160 joules centimeter square plus 5LA, 160 joules plus 5LA photodynamic therapy, and 5-ALA and necrosulfonamide with indicated concentrations. In summary, inhibiting G7 GSCs with NSA at indicated concentrations after PDT treatment suggests a potential involvement of necroptosis pathway in cell death mechanisms elicited by treatment regimen, as well as partial dose-dependent rescue effect after post-PDT treatment over a 24 hours time period. To further investigate the potential involvement of necroptosis pathway in GSC's post-PDT5LA treatment, Western blot analysis was conducted, and this analysis aimed to assess the protein expression levels and activation status of key components implicated in cell death events involved in necroptosis, as well as to elucidate the main differences with apoptosis pathways. Western blot images represent necroptosis pathway activation in G7 GSCs. This data from 3 biological independent experiments with phosphorylated MLKL expression in all timings post-PDT 5LA treatment, which is seen 20, 40, 60, and 80 minutes respectively after exposure. On the other hand, G7 were subjected to BH3 mimetic treatment targeted with BCL2, ATB737, and MCL1 S63845 inhibitors, showcasing apoptotic death events. Caspase 3 and 8 cleavage with cleaved PARB reduction can be seen and elucidates the primary distinctions. Jerkat extracts treated with cytochrome C served another positive control for cleaved caspase 3. One-way ANOVA analysis represents the significance of G7 GCs treated with DMSO, light alone, 5-LA alone versus photodynamic therapy 390 joules centimeter square plus 5-LA at indicated time points. The data was followed by 2 post-hoc analysis, and error bar represents standard deviation. The presence of phosphor-related MLKL at serin 345, which observed in this cell line and other cell lines observed following PDT5LA treatment in G7 GSCs, signifies the activation of necroptosis pathway, thereby precluding the involvement of apoptotic death events. What about clinical practice? The clinical relevance of necroptosis in cancer cells relies in its potential as a therapeutic target to induce programmed cell death, particularly in resistant cancer cells, and to modulate the tumor microenvironment and to enhance immunogenicity and improve treatment outcomes. However, despite all this successful pathway activation the main challenge remains to target GBM, as photodynamic therapy destroys GCs at 0.5 centimeter deep in surgical cavity at this wavelength, whereas tumor cells, they remain approximately at 2.5 centimeter depth resection. However, low influence rates, such as 25 joules centimeter square, allows more light to penetrate deeper into the brain tissue before being absorbed or scattered. This phenomenon facilitates deeper tissue penetration. So 25 joules centimeter square has less adverse effects for the brain tissue and deeper penetration, but how relevant it can be as it is known fact that it has no adverse effects for the healthy brain tissue. However, combining PDT5LA with reduced doses of small molecular inhibitors might overcome this resistance to apoptosis, although PDT5LA is effective in target, as I mentioned before, within 0.5 centimeter in surgical cavity, so its limited depth of penetration leaves deeper tumor cells, well, up to 2.5 centimeters from resection margin untreated. And the findings suggest that combining PDT5LA with small molecular inhibitors, as I mentioned before, with ATB737 and S63845 could overcome these limitations by sensitizing tumor cells to apoptosis, thereby extending the therapeutic reach of PDT. However, on the contrary side, the activation of necroptosis and parthenosis, as was shown, not only ensures the eradication of tumor cells, but also the potential to elicit the immunogenic response to the release of dumps during necroptosis, and this immunogenic cell death presents strategic opportunity to combine PDT with immunotherapy. Coupling PDT-induced cell death with immune checkpoint inhibitors or other immunomodulatory therapies could amplify the immune-mediated clearance of glioblastoma cells and induce those beyond and direct reach of PDT. Such approach could transform PDT to predominantly local treatment modality into a central component of, I would say, a multimodal therapeutic strategy for glioblastoma. Thank you for your attention. See you soon.